Well, good morning, and for some of you, good afternoon. Welcome to Beyond Compliance, the webinar series sponsored by SafetyChain. Today, we got a really cool, very timely, I think, topic as well: how same-day salmonella quantification guides intervention strategies and decreases costs. I think there's a couple of key results any one of us on the line could use a little bit more of. So. If you want to advance it, I'm Aaron Bolshaw. I'm with SafetyChain Software, and a little bit about the Beyond Compliance series. We meet monthly, and we have these great topics for the food and beverage industry that focus on, maximizing productivity, reducing costs, becoming more efficient in the way that we go to market, even with our food and beverage companies. And so today, I'm happy to be joined by our session sponsor, Hygiena. A little bit about SafetyChain. For anybody that is unfamiliar, SafetyChain software is the #1 plant management platform that does help to food and beverage companies increase yields, maximize productivity through safety, compliance, and production through their facilities. Really the only end-to-end complete solution for food facilities from that load in to load out. With over 1,500 facilities in North America and growing, we're quickly becoming the go-to and the gold standard. So with that, so you all can hear us, all right, but we can't hear you. But we do want to hear from you. And so to do that, when you have questions, I'd like to ask that you just use your question panel over in the webinar panel. Right, so we're going to get to those questions at the end. We do have scheduled about a half hour today. We may go over a little bit more if we feel like there's some really good questions. We want to stay on the line, but we do want to keep this timely, topical, and informational. So at the end of all of this, everybody's going to get the links to see the recording, see the information and the slides, all that good stuff, of course. And then if you're having any audio issues, sometimes just calling in, like maybe if you're on your computer and you're using your VoIP, calling in using the dial number will help you out there. So give that a shot if that's any problems. Okay, so with that, did want to talk a little bit about Hygiena, our session sponsor today. So this is really fun. So Hygiena is best known for ATP monitoring systems using PCR-based foodborne pathogen detection. Some of you on the phone may actually use the VAC system maybe already. I know some of our customers do. So, but it's about DNA fingerprint molecular characterization system. That's a mouthful. And then also mycotoxin and allergen tests, of course, really have the devices, the environmental collection devices, and much, much more. But I'm not going to be the perfect person to talk about this. I'd like to introduce the person that can speak a little bit more to it. I'm the software geek. So it's my pleasure to introduce everyone, David Luedeke with Hygiena. David, thanks for joining us today.
Yes. Hello, everyone. Thanks, Aaron.
Why don't you just, to start us out, why don't you give us a little background, where you come from, a little bit about what you do day to day and your expertise in this area?
Yeah, sure. I've got a few bullet points on the screen there for everyone can see it. And overall, I'm kind of a jack of all trades, master of none. Overall, not only in my career, but overall, I like to do a lot of things in general. But I do, my work experience in the food industry spans about 15 years. I have worked with NSF International, managing retail food safety programs. I spent a lot of time in the lab at a company called Battelle Memorial Institute, headquartered in Columbus. That was really some unique work with select agents and BSL-3 organisms. I worked a lot there on risk assessments conducted for DHS. And then my education is really in biology, master's undergrad in biology. I did also spend some time working for Burdock Group Consultants, looking at food ingredients, helping companies bring their new ingredients to market. So worked in different segments of the food industry and started working for Hygiena almost 3 years ago now. And so,
very good.
Moving to our topic of the day, Salmonella Quantification. Before I hop in, I've been happy to have a few conversations this morning with those of you that joined. I gave you a last minute notice. I know you still hopped on, so I appreciate you guys joining and everyone else that hopped on to listen here. It's a good format. I like the shorter webinar that Ann and Aaron put together for this, because it gives you a taste. It helps you become aware of a newer methodology out there, and then you can decide going forward if it's something you want to invest more time in or not. So I'll hop right into it here. We're talking about Salmonella Quantification, and I'll refer to it probably most often as SalQuant for short. I do have a picture here of the back system. I've got a smaller unit there, the X5. This really pertains to our larger Q7 unit. I'll be focusing on Salmonella and specifically poultry in this talk, but I don't want you to limit your thinking to that only. The methodology applies to other pathogens of concern. We've also done a lot of work with other food portions of the food industry, so pet food, beef, pork, not just the poultry. So I've got examples that focus in on that, but it's a much broader application than the examples that I've got in the slide deck. Also wanted to say that everything I've got contained here, and the overwhelming majority of the body of work we've put into this is all in partnership with the industry. The data I show has been provided by our poultry industry partners and guided by their requests and their interest in what we've got here. So going further along here, we're all aware of the need to stay on top of new technologies, as I've got in the title here. We've all got people monitoring what's coming out. We're going to conferences, networking. What are you doing? What doing? We're all aware of what each other are doing, looking for the newest and fastest and greatest technology. We know that regulations are driving a lot of this. We see changes in FDA and USDA guidelines that kind of guide us, may influence us. And what we've heard from our customers is the currently available enumeration technologies do not provide a rapid, actionable way to drive and help our decision making. That's what we've heard. We're aware of this need, have been, and what we sought to provide is a more reliable, accurate, precise enumeration method than what is traditional or conventional, or has been used for years. And I know even in the last year and a half to 2 years, I know people are interested in this, because I've sent out a few emails to folks and gotten a response within 3 or 4 minutes saying what we have here, are you interested? And they say, yes, please, can you come next week to tell us more about it, or can you tell us about it this afternoon? So I know there's interest in what I'm about to go into here, just based on my own experience here. This pertains to Hygiena's back system and the real-time Salmonella kit. It's not a new platform, a new technology. It's just another option that's available with what's currently been out there for years. And I want to spend a moment on the plot in the right of your screen here, because I think it sets up what I'm really trying to communicate here. And if you look at the x-axis, we have like a process flow from the poultry industry, going from the flock all the way out to a ground final product to the right. And the testing that's traditionally done is a presence-absence-based approach, or prevalence testing. And so on the right y-axis, you see your presence represented by a 1, absence 0. And so all along those test points, what's commonly done is you test to see if Salmonella is there, and the outcome is whether it's there or it's not. And what I want to focus on is what can be done to look at the concentration of Salmonella at each of those time points. So now focusing on the red line. and the left y-axis, the log CFU per mil or gram per sample, so that you not only know whether it's there or not, but you know how much Salmonella is there. You can see how much of a decrease or increase has occurred after your intervention or after each step. So the red line, the option to choose that path to quantify if you'd like, is a new approach that I'll be getting to here in more detail. So the current state, a lot of us may be aware of the most probable number method. This approach has been around for a very long time. I know there's a paper published in 1915 about the MPN method. It's based on serial dilutions. In the picture on the left there, I have a sample represented by the blue bag, or the blue liquid in the bag there. And samples are diluted to culture tubes and then dispersed to other culture tubes and then cultured on plates. And this approach takes time, and it's very cumbersome. There can be one or more periods of time of waiting overnight for results. There's a lot of waste. There's a lot of storage, shipping, and overall, if you run these before, you know it's prone to errors with so many manual steps being involved. And what I'm putting forth here is an alternative that seems to be just as good, if not better, than this gold standard that's been around for so long with the BACs and this PCR platform, where a lysate tube and a BACs tube can really replace all the tubes and plates that you see on the left. So a total of roughly 300 microliters or less. And in the workflow comparison here, looking on the left, an actual procedure that follows an MPN approach that can take 3 or 4 days. You see several time periods at step 4, 6, and 8 where you have to wait overnight. And then you use tables at the end to compute your concentration. On the right, we've got the SalQuant approach with an incubation period for 6 hours and a run on the backs of about 75 minutes, so a much shorter time to result. This approach with the backs does utilize a CT value. It's the cycle threshold. It's basically how many heating and cooling cycles it takes for the algorithm in the system to determine a positive or negative result. And so that's what we use to compute the Salmonella concentration. SalQuant, it does have a wide enumerable range. Some sample types are heavily loaded with bacteria. They have very high concentration, some or a lot less, or perhaps the concentration that you're interested in testing for detecting is perhaps very low. And what we found in comparing this approach to the traditional method is that the range is wider than running MPNs. There's also a limits approach that's been out for years with some other technologies and methodologies. It's more of a semi-quantitative approach where a threshold is established by an operation or a company, and it is set up such that if you get a positive result for that sample, you know you've exceeded that threshold that you've established, and then maybe you plan to divert that product to a cooking or another kill step. Or if it's not positive, then that sample proceeds to your established flow and SOPs. I have a lot of data. I like data, and there is a lot of data that we have generated for this effort, especially over the last couple of years. We've run a lot of different food matrices and generated a lot of curves. I call them curves. It's really a specific formula of time and temperature and mass and a few other steps that are specific to each food matrix that we test. But we've put in a lot of work to establish all those metrics, all those parameters for the different food matrices that we're working with and plan to work with in the future. We've done a lot of this work with Texas Tech University. And as I mentioned earlier, a lot of these curves, a lot of the development, almost every sample has been provided by industry, or we've run at the request or interest of poultry industry and where they believe the testing is going. The statistical parameters that this approach has shown through our data show a better accuracy and precision than using culture tubes, culture plates. It actually exceeds the conventional methods, basically much tighter error bars so that you can detect subtle differences at statistically significant levels, allows you to better optimize your interventions. Just another slide here. I won't spend much time here, but just the steps are fairly simple. It's just a bit of flexibility with the current BACS system and PCR kit setup. I've got here just some examples of the range of food matrices that we've worked with. There's on the left here from the live side or live production, we've got boot swabs, which are a type of environmental sample. We've looked at the ceca and viscera packs, which are internal organs. We've looked at feed samples, different types of feed with different additives, different recipes in the processing environment, different types of rinsates and skin. We know that skin has pores and that Salmonella likes to hide in those pores when they open and close at different steps, all the way out to the final product, a ground product. So in a lot of people may be interested in what's coming into your plants. You maybe or maybe not have a feel of what already resides in there, but what's coming in. And so thinking about that category on the live side, think about it in terms of what's coming in from a different location or a different facility or a farm or a batch, or not just what flock or what birds are coming in, but what if you could rank or sort by highest to lowest risk by knowing what the load is of those flocks or of those different batches that are coming to your facility. What types of adjustments would you make or interventions would you apply? So that's the type of information you can get from, say, looking at a boot swab or other samples from the live side. And I actually have some data, a couple slides here with some plots and some data that we've generated. And again, specific to poultry, a problem could be what is coming into your plant from live birds. And so using SalQuant to look at what concentration, what Salmonella load is coming in to your facility. As I mentioned, applying a pre-harvest intervention or adjusting or monitoring interventions are things you could do in getting results relatively quickly, the same day, as opposed to waiting overnight or a couple or 3 days. So in this example, on the left here, I've got some of the details of the experiment. This was looking at ceca samples. We compared SalQuant to a miniaturized MPN, and the SalQuant enrichment time, incubation time was 8 hours and plus 75 minutes to run on the back, so roughly the same day there. And the outcome of this experiment showed that the two methods were equivalent. The plot at the right shows the data points, the y-axis on the left, log 10 CFU per mil of rinsate of the ceca samples, And I know it's kind of a fancy whisker plot, but the two methods in this control versus treatment test using the all pairs Tukey-Kramer they were equivalent at the 0.05 level. So having SalQuant provide faster results using less materials, perhaps less cumbersome overall compared to a current method or a more traditional method, is something that's worth considering. Got another example here, and this is from the processing environment, looking at the effectiveness of an intervention efficacy. This was 5 samples per processing location. We tested both for prevalence and overlaid that with SalQuant quantification. This was using rinse aid again and a 6-hour incubation. If you look at the plot to the right and the left y-axis, you can see your prevalence rate. The 80% of your samples tested positive at rehang there. Moving to the right, at post-evisceration, 100% of your samples tested positive, and then at post-chill, there was no Salmonella was present. So prevalence testing shows maybe it's an increase, maybe the same number of samples had Salmonella and we missed one on the left at rehang. But if you overlay that with a quantification approach, looking at the y-axis on the right now, referring to the dotted line at rehang, the data shows roughly 1.2 log-10 CFU per 400 mils of rinse aid. And then moving to the right, it looks like an actual decrease, Salmonella concentration, and then moving further to the right, 0 at post-chill. So using statistical power with more samples, you can get even better data. But what if you had that ability to see what's actually happening? You know, what's your risk at now, knowing that your concentration in that load is trending towards being less? Maybe you would make slightly different adjustments, or your next step towards optimization would be a little bit different than if you just had your prevalence results. So this is really my last slide here for key takeaways, just to revisit some of the things I've run through, stated previously. But we've ran a lot of samples. It's ever growing. It's not just poultry, pork, beef. Anything that you can test for prevalence, you can test for the concentration. You can quantify. You can use SalQuant. You may not believe or feel or want or choose to do this type of testing for quantifying. Maybe you're just interested in just presence absence. We've heard that. But they've also said they might be interested in conducting a special project or a biomapping type assessment on a quarterly basis or maybe an annual basis. The statistical parameters show that there's better accuracy, better precision with SalQuant compared to a traditional method, and actually meet or exceed this conventional method. It does show some actionable data that comes faster than using culture plates or tubes. And the data shows that it's reliable, accurate, and precise. It's an enumeration method that is available to try now, to potentially even give you better results than you're used to getting with the method you've been using for probably years. And so I'm wrapping up with this. I know I've kind of run through it pretty quickly, but I believe we have a few minutes left for questions. So thank you, everybody, for listening. I hope everything made sense. And Aaron, you want to take it from here?
Yeah, absolutely. So I'll skip the jokes about me not paying as close of attention in chemistry in college as I probably should have. However, I think that this has been fantastic, David. And we do have some questions that have stacked up. So I'll just remind everyone, this is the last chance to get any questions in if you have them. I've got 4 so far. So I'm going to just kind of start at the top. Chelsea wants to know, how does the SalQuant kit give an exact value of Salmonella per CFU per milliliter, or a specific range, and she kind of says, i.e. 2.0 to 2.5 log. Can you speak to that?
Yeah, sure. We've run a lot of tests comparing different BACS units, different food matrices, different number of samples to really zero in on a very tight error bar range, like even close to 0.25 log CFU per mil in some cases. It really depends how many data points you have. It is a good question because it is that tight range that's much tighter than an MPN approach. So I didn't show, all the statistics on that last plot, but the data we've generated shows that those significant differences can be detected within a very slight increase or decrease.
Great. Yeah, that's you always want your confidence. There's a lot of folks that are on the line, David, that are definitely in the lab side, right? And so every tightening of any kind of spec like that, and just confidence that you can give into the testing scenarios means a lot. So she got another great question though. Do you have a validated matrix for environmental samples?
Yeah, that's another good and common question. We are pursuing AOAC validation for the matrices we're working with. We've got a few of those in progress, and they're basically lined up in a queue. I believe that validation is occurring for a couple, perhaps within this quarter. But currently that validation is not finalized, but it is in progress here. As everyone knows, it does take a lot of time. We're on a little bit faster path for AOAC validation for those, but we do have a lot of fit for purpose data that we can rely on.
Great. Thank you for that. Another one about the data, sort of a general question about, they say rendering. I think what they mean, I'm going to try and sort of re-encapsulate it. How is the data shown? So in the back system, after a test, how is the data presented, right? So, and that's interesting to me too, because I guess there's a lot of different ways you can do a lot of different things with the data, but can you just talk to maybe how, David, your customers, Hygienist customers generally see, use, and share the data?
Yeah, sure. That is another good question. So the basic output of the BACS is that CT value, and we started down this path on being able to export that CT value to a basic Excel sheet. And once we really got moving on that, we've got some things in development so that the software actually produces an easy concentration estimate based on the CT value. So it will be eventually contained within the software or an additional app, and then folks can share data however they decide to in the labs. Does that seem to answer the question?
I think so. Yeah, I think so. And, I gravitated towards that question because, as the software component to many production facilities, we geek out on data. We love it. And so, and it speaks to the importance of getting that in a data format that's shareable, reportable, so you can go back in time, you understand what's going on if you're audited, all those kinds of things. So, yeah, we've got a couple more questions, and then I think we'll wrap it up. So, Vicky's asking, is the SolQuant result reported as CFU/G or milliliters, or MPN/G or ml? So those are some, I hope you can decipher what I just read.
No, it depends on the sample type, the food matrix, whether it's a rinsate being tested, or if it's a rinse of a different matrix, or if it's a ground patty. That determines whether the result is CFU per mil or per gram or per sample, or as you saw, per 400 milliliters of rinsate. There's just some calculations behind there that can produce the concentration in the units that you're interested in.
Okay, very good. I'm glad you were able to decipher that. Two more questions, really good ones. Actually, this speaks sort of to the bigger process, right? If you've got this internal system, right, and you're doing your lab tests internally, would an external laboratory still need to be used for periodic validation of this method for FSMA purposes, FSMA?
I think that each will determine what they're comfortable with regarding the data that supports the method that they choose, using data that we've generated, what's going to be AOEC validated, what a third-party laboratory has or can do for them, or what they can do internally, or what we can produce for them. So I see a wide range currently of our customers that are using this approach already. There's a wide range of data that they have to support the direction they're going.
Okay. Yeah, that's a great question. I think that I'd ask, we're recording this, the SafetyChain side, we have some solutions architects and some experts in FSMA that we'll follow up with you as well on that question. That's a healthy question. It speaks to, do you want to add another level of that risk mitigation? And a lot of companies, I think, will. And especially as far as auditing goes, then you just get into a whole ball of wax. So great question. And then one more question and we'll wrap it up. David, I think if you go to the last slide here, there's a couple of places for some folks to find some information, definitely on SalQuant. Visit Hygiena.com. There's other resources available on SafetyChain.com. Won't go into that, but there's definitely some more resources as you want to and need them, everyone. Last question on matrices. Is there a cutoff of moisture content of the samples for appropriate results?
That's a good question. I can really only speak to the matrices that we've tested and different feeds types that we've tested. We've been able to navigate how to do that. There's been some really unique matrix types that we've worked with, and they seem tougher. There was maybe an obstacle or two that we faced, but we were able to come up with the right formula, so to speak, where we did find the right protocol for volume or mass, time, temperature, whatever it may be. We haven't come across a matrix yet that we weren't able to produce a curve for and use this approach with.
Very good. Very good. All right, that's it. We've got a lot of many smarter people on the phone than I am, you included. So, David, one, I just want to thank you personally. This has been fantastically educational for myself. We've been working on this for a few weeks. You walked me through the entire poultry process. In the process, we found out, we have some same customers, and there's just a lot of really great things that Hygiena is working on. And so happy to have you aboard. Maybe down the line we'll touch base again. But that's it. Everyone, we're going to wrap it up. This has been Beyond Compliance for June 2020. We hope you're all staying well out there, and we look forward to seeing you again next time.
Thank you, everyone.
Thanks. That ends the show. Take care, everyone. Cheers.